Yesterday we talked about osmosis, well, facilitated diffusion first, and the channel and carrier proteins that allow for the diffusion of particles that have charges or that are larger in size and cannot simply pass through the cell surface membrane. There is specificity to these channel and carrier proteins. It's not that it's you know a route for any particle to move through. Channel proteins are sometimes gated, so they need some sort of signal to either open or close. And close. They could be voltage gated or ligand gated, so they need a potential difference or some sort of other molecule or particle binding to cause the opening or closing. Carrier molecules carry particles across cell surface membrane, rotating and changing shape while they're at it. And all of this is diffusion, right? So it's moving from a region of high concentration or high electrochemical gradient or high pressure or whatever it is to a region of low concentration, and it does not utilize energy from the cell, so no ATP basically is used in the process. Then coming to osmosis, this is a special case of diffusion which is for water molecules, and it is the movement of water molecules down a water potential gradient, so going from high to low through a partially permeable membrane, right? So that membrane can allow water to freely move through, but is selective for other solutes. Right? So the solvent can pass through, the solutes are selective. It's selective for the solutes. Right? And so that way particles can accumulate on either side, and that affects the water potential. Right? So we talked about free water molecules are not, so what affects osmosis is the free water molecules. Right, the ones that are not associated or interacting with any of the solute particles. The greater the free water molecules, the greater the water potential. So that's one thing to consider. The other thing is osmotic concentration, which is the concentration of solute molecules that have an osmotic effect. Right, so such as ions or polar molecules, particles that dissolve in water. Right, so molecules such as lipids or starch do not have an osmotic effect, and so they do not have, they do not change the osmotic concentration of the cell or the tissue fluid or the blood or wherever they are, because they don't, like lipids are hydrophobic, starch doesn't dissolve, it doesn't interact with water, and so it's not taking part in effects on osmosis. And so you put these two together, if you have a solution that has a high solute concentration, so that's called hypertonic, and that means it has a low water potential because the lesser free water molecules are there. If it's isotonic, that means that the solute concentration is the same as whatever other side that you are comparing it to, and so there's the water potential is also the same on both sides, and so nothing's happening. And a hypertonic solution has a low solute concentration, therefore a higher water potential. So what happens to a cell, whether it's a plant cell or an animal cell, in an isotonic solution? The solute concentration inside and outside the cell is the same, and so is the water potential. And so there's no net movement of water molecules, meaning that there is movement on a molecular scale, because molecules are vibrating, they do have kinetic energy, and so water molecules are going across the partially permeable membrane of the cell membrane, whichever membrane we are talking about. Possibly the epithelial cells of the capillaries, but right, there's no net change. So if 10 are moving one way, 9 or 10 are moving the other way, it's pretty much, it remains constant. Now talking about animal cells, if they are put in a hypertonic solution, so there's more solute concentration outside the cell than there is inside, and that means that there is a lower water potential outside the cell than there is inside, and so water will move out. It will move out of the cell. Cell will shrink. That shrinking will cause the contents to become concentrated. Right? So contents become concentrated, and obviously that affects metabolic processes, and the cell will end up dying if it stays that way. Uh, it is detrimental even if it happens for a short time, but you know, recovery may be possible, but it will lead to cell death. A hypotonic solution, the water moves into the cell. Why? Because there's lower solute concentration on the outside. That means there's a higher water potential on the outside. Uh, right, and so water is moving into the cell. If it continues to move in, the cell will first expand to a certain limit, not too much. So let's do it this way. It expands or swells. Well, actually, I did write that. And then, if it doesn't stop, then the cell will end up bursting because it does not have a cell wall, and so there's nothing keeping it in place and giving it physical strength. Now, today we will talk about plant cells, and so there's a bit of detail to go with the plant cells in both cases of hypertonic and hypotonic solutions. So plant cells, let's do hypertonic first. So if a plant cell is placed inside a hypertonic solution, the same concept applies, right? Water will move out of the cell. That is what will happen, obviously. Now, the large central vacuole in the plant cell is one of the main stores it has. It's filled with water. There's water in the cytoplasm, obviously, but there's a lot of water in the large central vacuole in plant cells. So the large central vacuole will shrink. Right, and right, water also leaves the cytoplasm, obviously. And so what happens is that the protoplasm, so that's all the living material, that's the cytoplasm and organelles and everything, that shrinks overall. And what happens is that the cell membrane, because just a recall, the cell membrane is partially permeable. The cell wall is fully permeable. Right? So the cell wall of the plant cell does not regulate the passage of any materials into and out of the cell. It's basically there for structural support. So the cell membrane, right, also shrinks. And starts to pull away from the cell wall. Right, the point, right, just when the cell surface membrane is about to pull away from the cell wall, it is called incipient plasmolysis. Incipient meaning bound to happen, right, or just happening. Plasmolysis is the state where right, the cell contents have shrunk due to loss of water. Okay. Now in all of this, the cell wall is not affected at all. Right? Why? Because it is made of cellulose and it is a rigid structure, and the whole point is it gives mechanical support. And because of that, even though the cell content is shrinking, the size and shape of the cell is maintained. Why? Because of the cell wall. So you'll have something like this. And if I just change colors, you've got the cell wall. It's a typical rectangular, and you may have the cell wall, right? It's pulling away. And the vacuole inside, so that shrunk. That's the vacuole, the nucleus, right? However, that's affected. And so, right, this is right incipient plasmolysis or plasmolysis that has just begun because it is pulling away. Right, obviously, I'm not going into detail with the other organelles. And so, even if it shrinks, the cell wall is not moving, and so it's going to stay in place. What else do we have about plasmolysis? Now, when doing this in a lab, uh, right, what they do is to study the cells under a microscope, and they decide, at what concentration so osmotic concentration does incipient plasmolysis occur. And for that, they look at the cells and they determine, oh, well, uh, they write 50% of cells are plasmolized, so they've gone through the whole thing, they've completely shrunk, and 50% are not plasmolized. And so when they look at that, that is the point that they consider as incipient plasmolysis. When does incipient plasmolysis occur? Because you've generally, right, if you talk about osmotic concentration, you already put it into a hypertonic solution. Right, so it's not the concentration exactly, but at what point or how quickly, right? So whatever it is that you're measuring, with regards to the concentration of the solution, it is hypertonic, but how hypertonic is it? And so when does it occur in that particular solution? When they notice, right, it's 50-50. Now, what happens when they are completely plasmolized? That is when the right cell surface membrane completely pulls away from the cell wall, and the protoplasm shrinks, and it just becomes like a concentrated mass. So cell wall and protoplasm has shrunk. The cell is now completely plasmolized, right? Or it has suffered plasmolysis. Any questions?
No, sir.
Right, once I explain the next one where we're putting a cell into a hypotonic solution, I'll show you some images from the book.
Sure.
So you've got hypotonic solution for a plant cell, and so obviously water is going to enter the cell. And the cell will swell, just like with the animal cell, right? It will increase in size. So uh, the majority of the water is going into the vacuole. To the large central vacuole of a plant cell, right? And that expands in size, and that pushes the contents against the cell wall. Remember, the cell wall is not affected, it is rigid, it gives mechanical support, and so that is going to play a very important part over here. Because, right, just like the animal cell, the plant cell did suffer plasmolysis, right? It shriveled up, but on the inside, its shape was maintained because the cell wall. When a hypotonic solution, contrary to the animal cell which burst, the plant cell is going to survive. In fact, the condition we're going to talk about in the plant cell, that the condition it achieves in a hypotonic solution is the condition that it's normally in. So what happens when the contents of the cell are pushing against the cell wall? The cell wall, right, exerts a reverse pressure. Right, so this expanding and pushing against the cell wall is due to, right, hydrostatic pressure. Because of that, uh, but sorry, let me go back on that. Mixed it up. So this in turn, right, the cell wall produces an inward pressure, a reverse or inward pressure in response to the contents pushing against the cell wall. And that is called the hydrostatic pressure. And what it does, it sort of cancels, or it starts canceling out the tendency of water to move in. So, right, it negates the tendency of water moving in, even though it's hypotonic cell. And so this is the point where water will actually stop moving into the plant cell, right? And so what happens is that a balance is achieved, or balance is reached, right? Where the water potential of the surrounding solution and its potential of wanting to move into the cell becomes equal to what is called a pressure potential So we talked about, diffusion being due to the electrochemical gradient or a pressure gradient or something like that. And so this is a pressure potential that is being generated by the cell wall due to the hydrostatic pressure, the push of the cell wall back onto the cell contents. And because it's pushing, you could think of it maybe like, you know, I would say a balloon, but you know, it's not going to like a balloon. I think that's the same example they give in the book. So it's like a balloon filled with water or jelly, right? But it's in a box. So even if you're filling in water, it's only going to expand, right? Even if it has more elasticity to it, it's only going to expand up until it's completely touching the walls of the box and the box is rigid. And so like up until that point, if you're adding water, the water will continue to go into the balloon. The rubber is elastic, it will continue to expand. But at the point where it's completely touching the walls of the box, it doesn't have any more room to expand. And so water will stop filling in the balloon, right? Even if you're adding more, if you're forcing it in, it will just spurt back out. And so that is a good example to understand what's happening over here. And so a hydrostatic pressure by the cell wall, right, going back on the cell contents, and right, so a pressure potential is created, and that balances out or equals the water potential or the tendency of the water to move in. And now, right, the plant cell is swollen, or it is, right, called turgid. That's a term for it. And right, the state is known as turgor. And right, this is actually important for the plant cell because it, gives it structural support. So water is important for plants. Well, this is one reason, because they take it up. All the other processes that they use it in, sure, but right, most of the plant cells, the stem, the leaves, they're actually in a state of turgor. They're all turgid, right? And that's what helps especially the stem plants, right? Because they don't have, the outer covering of the bark and the thick woody, right, structure of their stems or trunks. And so the stem plants, or the green stem plants, right, it is turgor in the leaves of the stem that actually keep them upright. Any questions? I will take that as a no. Okay, I will take you to the book for the illustrations. So here you can see, right, this is a red blood cell. A is, right, the cell is burst, so it is in a hypertonic solution. B is the cell is just fine. It is an isotonic solution. And right, C is, wait a minute. Right. Okay, yeah. And then over here, right, you've got plant cells that have undergone plasmolysis. These ones are, right, in a state of turgor. Okay, and with that we are done with osmosis, and so we will move towards active transport. Where is with the white background? Okay. The next step is active transport. An active transport is the movement of particles against or up a concentration gradient which means they're going from low to high, and right, that's not something that would happen on its own, and so in this case energy is provided by the cell in the form of ATP. Right, so ATP is hydrolyzed. Right, what you get is ADP, inorganic phosphate, and a bunch of energy that is used for the movement for active transport. Now, before going into a bit more detail of the process, whenever you come across the word active or actively, any process that's happening actively, or it is an active process in the body, know that it is utilizing energy. In most cases, it would be ATP. But it could be, for example, in photosynthesis, in oxidative phosphorylation in the mitochondria, so that's respiration, and so it's using energy from electrons, right? But anyway, an energy input is required of some sort. So just keep that in mind. If you come across that, okay, right, energy is needed. Now, active transport, right, is a process that is happening in a lot of places in the body, right? A very marked example is the absorption of the products of digestion. So nutrients by plants maybe, or products of digestion in the small intestine, whichever works. Products of digestion, and so right, if you leave it to diffusion, there's a high concentration in the lumen of the intestine, and they're taken up by diffusion into the microvilli and then the bloodstream, and sure, diffusion gradient is maintained, right? But then there would come a point where, for example, you've got some left over there, and so active transport plays a role, and takes up the maximum possible, maybe not 100%, but as close as possible, right? It's also involved in maintaining a potential difference, so, or an electrochemical gradient in neurons, and that is important for nerve impulses to fire, right? So active transport is plays a very big role over there. How does it happen? So, right, it happens using carrier proteins, not channel proteins, but carrier proteins. Right, and uh, right, the enzyme ATPase is involved, and that is what catalyzes the hydrolysis of ATP. Now, depending on where this is happening, if it's happening on the membrane of a cell, if the cell is taking something up, then the ATPase complex, right, will be membrane bound, so it will be just there. So it's able to provide energy to the carrier proteins. Right, and because it's occurring across a membrane, so it's in most cases that is how it would be. Right, these carrier proteins are generally membrane spanning. We've talked about how they work. And right, this works. I mean, it could work, right, for movement into or out of the cell. Right, so in the case of absorbing nutrients or products of digestion, right, they're being taken up into the cell. But in the case of maintaining a potential difference, what actually happens is sodium ions are actively pumped out of the neurons. Right, so it could work either way. It's not like, oh, it only happens when uptake is required. It could be removal of certain ions. And for some reason, right, the entire system is, right, also called, this is mentioned in the book, so I'm mentioning it here. It's also called a gated system because they say that it acts as a gate. Where it go? Yeah, it acts as a gate and controls what comes in and out of the cell. So that bit, are we missing anything? We're talking about the neurons, so that's detailed, you can go over. And yeah, so just remove that bit from here. And so at the end, right, it is a combination and balance of diffusion, right, which includes facilitated diffusion, facilitated diffusion, osmosis, and active transport that keeps metabolic processes running. Oh, and one more thing, right? Active transport is not necessarily just happening at the cell surface membrane. It could happen within the cell as well, right? With organelles, so on a more localized level, for example, the Golgi apparatus or the endoplasmic reticulum or any other organelle, right? Any other membrane-bound organelle. Let's add that because you need a membrane for active transport. Right, in fact, you need a partially permeable membrane for active transport as well. Right. Any questions? Anything? So I'd like to wrap this up with the final bit that talks about uh, endocytosis and exocytosis. And that is basically a bulk movement of materials into and out of the cell. So endocytosis and exocytosis. Right? Endo means into, exo means out. So, right, these are two processes for bulk transport. Right, so they can work for either very large molecules for which membrane proteins are not enough, or a large quantity of molecules. Or let's say particles instead of molecules. So an example as large as it gets of endocytosis is a white blood cell, right, performing phagocytosis and taking up an entire bacterial cell. And so what happens is that the cell membrane stretches. You've got the bacterial cell over here, poor guy, and it stretches and starts engulfing the bacterial cell, and then these two ends meet, and so the bacterial cell is now inside a vesicle cell. So the bacterial cell is not like, it doesn't become part of the cell, it's not freely moving, so it is isolated from the protoplasm because it is taken up, right? It is engulfed in a membrane, the membrane pinches off, right? And so it's isolated within the white blood cell as a vesicle, and then you've got other vesicles that come in having lysozymes, so those are digestive enzymes, and they fuse with the membranes of this particular vesicle, release their enzymes, and digest the bacterial cells. So that's one thing that's happening. That is an example of endocytosis. You have an example of exocytosis where the cell might be releasing hormones or enzymes, right, in bulk. And so again, those are in vesicles in the cell. They move to the cell surface membrane, right? The membranes fuse. So the membrane of the vesicle and the cell surface membrane, they both fuse together, and right, that way the contents are released outside the cell. And then there's like on these lines, there's a process called, if I am pronouncing this correctly, penocytosis, which is a variation of endocytosis. And not surprisingly, it is a variation because here, right, the cell is taking up cytosis, and so the cell is taking up fluid from the surroundings, so it's taking up tissue fluid. Right, that's why it's called penocytosis or cell drinking. And why is it taking up tissue fluid? Because the tissue fluid has nutrients that the cell needs, and to fulfill its requirement, right? So active transport diffusion might not be as effective as just taking up the tissue fluid and then utilizing those nutrients directly from whatever is taken up. And is there anything else? Right, and so at the end they say that all of this is only possible because the cell surface membrane has the fluid mosaic nature, right? It's not rigid, it's got the phospholipids and the cholesterol and the proteins and everything else, right? And it is fluid, so stuff is moving about, and yeah, and that brings us to the end of this chapter, which is chapter 2A4 actually, so I didn't highlight that. So we're done up till chapter 2A4, and right, so next time we will move on to a new chapter and a new topic. We're done with membranes and transporters. Hello?
Yes, sir, I'm listening.
Okay. You good, by the way?
Yeah, yeah.
Okay. Right. So yeah, that's all for today, and I'll see you next time, inshallah.
Sure, sir. Take care. Allah Hafiz.
Allah.